This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 34, recorded December 12th, 2011. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
How are you, Dickson?
I'm well.
Do you know we are at 12-12-11 next year? It'll be 12-12-12.
And where were you on 11-11-11?
I was right here in my office, actually, on a phone.
Good.
I was on a phone call, and then later I emailed the person I was speaking with,
Right,
and said, you know, we were on the phone at 11-11-11 at 11, but not at 11-11 a.m.
No.
Dixon, I haven't seen you in a week. Where have you been?
I've been south. I travel south.
Is that a metaphor for being sick?
No, it's like, I ask little kids a lot of questions when I do outreach, and one of my favorite questions is, why do ducks fly south in the winter? Do you know the answer, Vince?
Because it's too expensive to walk.
It's too far to walk.
It's too far to walk.
It's
right.
too far to walk. That's
One
right.
of those two.
And the little kids groan and moan, and then they say, "Oh, I knew that." You know, it was kind of fun. So I went to Andros in the Bahamas to fish for bonefish.
Now, long-time listeners of TWiV and TWiP will remember
They will.
at another time Dr. Despommier went to
true.
Andros.
I do this periodically.
The name conjures up androgyny.
Well, Andros, I don't know where it derives its name from, to be honest. All of the islands have remarkably unique names, like Eleuthera.
Eleuthra.
Yeah,
Did you have a good time?
I had a great time.
Are you rested?
I feel rested.
All right.
You don't have to shake me to wake me up, but I feel...
Very good. It's good to have you back.
Thank you.
It was very quiet without you last week,
Well, of course.
and we did miss you.
Well, I felt...
But don't cry too much.
For me, Argentina.
The same thing went through my head as well.
I've been down there, too,
That's
by the way.
a famous line, isn't it?
Yes, it is. I've fished in Argentina as well. It's been a remarkably fortunate life that I've led so far.
Well, it's going to take a downturn now?
No, of course not. I hope not.
Well, here we
Sincerely
are.
hope.
The day you get back, actually, you got back yesterday.
I did.
It's Monday.
Yeah.
We are recording a TWiP.
This is our favorite thing to do.
Yes, we like recording. And
do.
I thought I would grab Dickson as soon as he walked in the door and get him to commit. Otherwise, who knows? He's so busy.
No, that's not true.
You are busy.
Never too busy for this, Vince.
Today is episode 34. We're going to talk about two papers.
We are.
And there is a theme running through these two papers.
There is.
You know what it is, Dickson?
I do. Endosymbionts.
Yes, and one in particular, right?
It's a Wolbachia.
Wolbachia and parasites.
Yes.
We have talked about Wolbachia and viruses on This Week in Virology.
In the beginning of our TWiPs,
Yes.
we discussed Onchocerca volvulus.
Right.
And we talked about, briefly, the Wolbachia associate.
That's right. And in terms of virology, if you're interested, TWiV 147, we talked about using Wolbachia to prevent dengue spread.
We did.
Wolbachia is an endosymbiont, you said.
It is.
What does that mean?
It's an obligate intracellular symbiotic organism.
Symbiotic. That means
It
somebody is
doesn't-
getting help,
Someone is getting help.
but not
from
necessarily both.
it. No, both.
But then it's a mutualism, right?
That's right.
Okay. The symbiosis is when at least
It's-
one of the two get something good.
No, it's
Or
a
is it
general
both?
term. Symbiosis is a general term from an ecological perspective that just means that two dissimilar species are interacting.
Okay.
But it doesn't necessarily say if it's a good interaction or a bad interaction.
Right, so all the other definitions fall within symbiosis, like mutualism,
Parasitism,
right? And anything else?
commensalism.
And what's commensalism?
It means we don't know what either one is doing for the other, and perhaps neither one is doing anything for the other. It's a neutral relationship.
You know, I hate to say this, Dickson.
Please.
We did discuss this previously,
I know we did.
but I do like reinforcement. I do.
I will give you a good example of mutualism, but it's not a symbiotic mutualism.
Are we symbiotic?
We are. No, we're not. We're not
No,
symbiotic.
because we don't live within each other, right?
No, we're not different species. You have to be different species.
What do you call people who help each other?
We call them friends. Lawyers, bankers, we call them family.
No, the lawyers are not friends.
We call them family. Some are.
The
Some
bankers
are
certainly not friends these days.
Some are. Now, you can't. I don't want to make a blanket
So
statement.
symbiosis has to be two different species.
Yes.
So people who help each other.
They're
are
symbiotic.
friends.
They could be symbiotic.
And we loosely use that expression to describe our interactions. We have a symbiotic relationship.
That's right.
But
That's right.
it's not in the bio or ecological sense, as you would say.
What I would say that we have together is that we've combined our scientific knowledge base from virology, immunology, and eukaryotic parasites to produce a synergistic relationship between us and the audience. So we can draw upon our vast, maybe from an age standpoint, at least from my perspective, our experience, and share that with people that are just starting out. And I think that's perhaps why we've been so well received by the general public.
We have 128 years of combined experience.
That's amazing, Vince.
I look at it more like 0 + 0 =
That's amazing.
0.
Well, yeah. To put things in another perspective, I just recently joined the American Chemical Society, perhaps maybe...
Recently?
I can tell you how many years ago it was. It was 4. How do I know it was exactly 4 years ago?
Because you just renewed your membership.
I did, and what do you get when you do that?
A mug.
And what is on the mug?
A periodic element.
Yes, and it's an order.
Not a periodic
So
element.
the very first one, if you join, is hydrogen.
You've gotten 5 so far
No,
now?
4. And the next one you get is?
Helium.
Correct. And the next one you get is?
Hydrogen, helium. Which? Lithium.
Yeah, lithium. And then...
My son knows this by heart.
That's right. And then there's the next one.
Beryllium? No. I can look on my phone. I have a periodic chart.
It's beryllium.
Then why did you say no?
I didn't say no.
Hydrogen, helium, lithium, beryllium.
That's right.
By the way, I have a wonderful periodic chart for my phone.
Excellent.
It's put out by a chemical company, EMD. Well, I'm not making this a pick of the week, but hydrogen, helium, lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, neon, sodium, magnesium. It rhymes. We should make a song out of it.
I think we've been scooped on that one.
Sorry. Hey, we digress, dude. What did we do here to lose the thread?
I don't know, but I know that I will never make it to the 92nd element on this chart.
Nah, you won't.
Nobody will.
And I don't think you want to either.
No, you don't.
All right, back to Wolbachia, intracellular symbiont. So the Wolbachia lives within the cells.
And not all the cells.
Well, what species
Of what?
does it live within?
Mostly invertebrates.
Yeah. So insects.
Yeah.
Crabs.
Sure.
So, what was the number? 60 to 70% of all invertebrate species or insect species? I don't
No,
remember
invertebrates. Invertebrates.
have Wolbachia in nature.
Yeah, because we're going to discuss two completely different groups of invertebrates.
Yeah.
One of them is a nematode invertebrate, and the other one is an arthropod invertebrate. And they both have Wolbachias.
And it does different things in each one, doesn't it?
Boy, don't it.
All right. I thought it would be interesting to do a Wolbachia for TWiP.
Yeah.
Especially since these do different things.
Sure. Now, in non-parasitic relationships, although I'm trying to think of what that might mean to the general audience, there are things called parasitic wasps that actually use ichneumon wasps are particularly popular in that group. They have these very long ovipositors, and the female sticks the egg into a grub of some other thing, like a beetle, for instance. I know you just have a lovely adult beetle on your desk here. Perhaps you want to put that up on the website to show you what I was pointing at.
Don't kill it.
It's already dead. It's a picture of a beetle. It's not actually the beetle itself.
Right.
But this ichneumon wasp species, all of the ichneumon wasps apparently have Wolbachia associated with them, and the Wolbachias, if I'm not misquoting this literature, because I used to be more familiar with it than I am right now, actually determines the sex of the invertebrate. It can actually drive sexual maturity towards femaleness.
Well, in mosquitoes, it does.
Yeah.
it causes a reproductive distortion to increase the reproductive success of females.
See that?
So if you took a mosquito with a female mosquito with Wolbachia and introduced it into the wild, that would drive its spread through the population, assuming everything else was neutral.
Right.
Now, we know that Wolbachia can affect the lifespan of some mosquitoes. And we also know that it can reduce viral replication in mosquitoes.
Yes.
And in fact, in particular, dengue virus.
Right.
And that's the basis for controlled release of Wolbachia-containing mosquitoes in Australia, which was done this year and which is an ongoing experiment.
Yep.
That was very successful in that the mosquitoes into which they had introduced Wolbachia, in the germline, of course, were able to spread within 2 small towns in northern Australia.
Yes.
And the mosquitoes formed a very high percentage of the wild mosquito population. So the next step is to go and put these into areas where dengue is endemic.
Correct.
And see the effect. So
Sure.
antiviral effects of Wolbachia.
Wild. Because
And how it does that is not known. It's
well,
very interesting.
exactly right. Exactly right.
But today, we talk about 2 parasites
Yes,
whose interactions with Wolbachia are quite different.
that's right.
Right.
But they're both vector-borne parasites, which makes them interesting.
All right. What are they, Dickson?
Well, one of them is a filarial worm called Brugia malayi.
Did we talk about Brugia?
We mentioned it along with Wuchereria bancrofti.
Wuchereria.
We emphasize Wuchereria because it's a more widely spread parasitic infection of humans, but Brugia malayi is also an infection of humans.
Mm-hmm.
And it can also infect other animals. And so
What does
it's
it
a
cause?
better
What does it cause when it infects us? Remind us.
Well, Wuchereria bancrofti could lead to a blockage of the lymphatics and then an elephantiasis-like syndrome.
Right.
And Brugia malayi is not as widely recognized as a causative agent for that condition.
But why do we study Brugia malayi then?
Because it's harder to study Wuchereria bancrofti because of the Koch's postulates involved in not being able to find an animal model suitable for a parasite that's so well adapted to humans that it won't infect anything else.
That sounds like an easy way out of things, doesn't it?
Well, there's no good animal model for Wuchereria, but there's a great animal model for Brugia malayi. So we've chosen the Brugia malayi model to look into the nuance with regards to how Wolbachia might affect the outcome of the infection.
So Brugia and other filarial nematodes?
Yes, I might add that Onchocerca is also a filarial nematode, and unfortunately there are no good animal models for Onchocerca volvulus.
Mm-hmm.
There are some Onchocerca that do infect other animals, but they don't exactly result in the same kind of pathology, and so they're not very good models for onchocerciasis.
So you can grow Brugia
Yeah.
and culture.
Yeah, you can. Well, you can obtain it in animals.
All right, and so that's how they do these experiments.
Some of them.
But you can't do that with Wolbachia. Not Wolbachia.
With Wuchereria.
Wuchereria.
No, you have to
I
use-
said Wolbachia because I can't pronounce Wuchereria.
It's quite all right. W.
You
bancrofti.
can't grow W. bancrofti.
No, and it's hard to get the adults because it's only in people.
All right, so Brugia malayi and other filarial nematodes have Wolbachia in the wild. So if you go out and collect specimens, you will find Wolbachia in them.
That's right.
All filarial nematodes?
Apparently so.
Really? Of all the parasites we talked about, just the filarial nematodes have Wolbachia in them.
Seems that way.
Is that because they're related to arthropods?
No, it's because they're transmitted by arthropods.
Ah.
And I think that's how they acquire these bacterial infections, during their developmental stages in the insect.
Aren't other parasites transmitted by arthropod vectors?
They are.
They didn't get Wolbachia, did they?
That's right.
Oh, so much for your hypothesis. No,
Well-
but you're right. It could have gotten them that way originally. But
Yeah, that's
for
right.
some reason, the filarial nematodes kept them, which tells us that they must be useful, right?
It didn't keep them. We selected them with our immune system.
You think?
I know.
Who's we?
Humans and other primates.
But
So the ones-
What's the evidence for that, Dickson?
Well, because the ones without Wolbachia, as we'll see, don't do so well.
Okay. Now, in the wild, what? Not in the wild. Excuse me.
That's
What
right.
is the experiment that has been done to prove that Brugia requires Wolbachia?
Well, Wolbachia is a bacteria, right?
Yeah, it's an endosymbiotic alpha proteobacterium.
Correct.
Did you know that?
Well, I do now. The proof of its necessity in the life cycle comes in a negative experiment. You have to eliminate it to find out if it makes a difference
Very
or not.
good. How would you do that?
Well, if it's a bacteria, Vince, there are antibacterial agents called antibiotics.
Here you go.
And so you'd probably try to use one of those antibiotics to get rid of it and then see what happens to the infection.
So you wouldn't expect the antibiotic itself to hurt Brugia
No, but
malaria, right?
That's right. But I want to inject a moment of historical importance.
Okay, sure.
I have a dear friend who I'm going to make the assumption that he's still alive, okay? Because a lot of my dear friends are not still alive. But this guy, his name is Bill Kozak.
Kozak.
Kozek.
Okay.
Kozak.
All right.
And
I'll give you that.
For many, many years, he worked on trichinella.
As did you.
As did I. And he became interested in its ultrastructure, as did I. And he was a very, very good electron microscopist.
Okay.
And he began to explore the anatomy of nematodes, particularly trichinella first, and then, Vince, he switched to filarial worms. When he did that, in 1977, he discovered these strange-looking inclusions in the lateral cords, as they're called. The nematodes have a nervous system, and they have one dorsal, one ventral, and two lateral nerves going on either side of the worm. And in the lateral cords, which they're called, he found these weird-looking inclusions, and he published this paper with some of his colleagues. By that time, he was at UC Davis, and they turned out to be Wolbachia.
Did he find that out, or did someone else?
Nope, he did.
How did he figure that out?
Well, he compared them to all the other endosymbionts that had been recorded. And, you know, the electron microscopy technology was just emerging into the daylight. I mean, the Philips 300 microscope was around for not much longer before that. The ultramicrotomy was improving using diamond knives and things of this sort. And Bill is a classically trained parasitologist that got interested in ultrastructure. And in fact, he even applied scanning electron microscope studies to the specimens to show that they were indeed independent units and not some part of the worm. So, bingo, look what it turned out to be.
So it says here, Dickson.
Yes.
Talking about Wolbachia, their presence is essential for the worm, as tetracycline-mediated clearance of bacteria from Brugia species leads to developmental arrest in immature stages and reduction in adult worm fertility and viability.
Correct.
So you treat the worms with TET, gets rid of the Wolbachia.
And something bad happens to the worm.
So the worms basically die, right?
Yeah, that's right.
Or they don't multiply any longer. That's it. That's the end.
Exactly.
So why don't we treat people with filarial infections with tetracycline?
Well, tetracycline is a pretty toxic antibiotic, for one thing. And the second thing is it's pretty expensive. And the third thing is, of course, it's pretty toxic. And the fourth thing is it's pretty expensive.
It says here you need several weeks of treatment to get rid of the filaria also.
And what's the downside to that, Vince?
Resistance.
Not only that, what else? You're going to get rid of all your normal microflora.
Oh, I forgot about those.
Like all 1,500 plus species in your gut.
Well, you're going to alter it.
Yeah,
You
but
won't get rid of it. Altering is the problem. Then undesirable ones predominate.
you don't
do
give this kind of therapy for that long a time.
So that's actually the topic of this first paper.
Yes.
Which is published in PLOS Neglected Tropical Diseases.
Neglected.
I think this may be the first PLOS NTS paper we have considered.
Great.
The name of the paper is Targeting the Wolbachia Cell Division Protein FTSZ. FTSZ as a new approach for antifilarial therapy. Now look at this, Dickson.
Okay.
The authors are from New England Biolabs.
That's interesting, isn't it?
It's a company that makes enzymes.
That's right, but they've had a rich history of dealing with worm infections, too.
Is that right?
Yeah.
Why is that? What's
do
the
a lot of
origin?
people up there? I don't know. They just sort of gravitated towards that area.
Interesting.
Yeah.
Then also the Skaggs Institute, the Worm Institute.
The Worm Institute. Vince, there's a whole institute devoted to worms.
That's nice. And the Scripps Institute.
Yeah.
Wow. I didn't know there was a Worm Institute.
Well, with Xenohybridus elegans, you know.
Yeah, that kind of worm. So this is a paper exploring a different target in Wolbachia than an antibiotic, using something else than the existing antibiotics.
Yeah, well, they're going to use antibiotics, but they're unusual antibiotics.
You could call them antibiotics.
They're plant alkaloids.
And derivatives thereof.
Correct. And they've got complex cyclical structures.
The idea being we have to get around the use of TET and other existing antibiotics.
Doxycycline.
Yeah, we don't want to do that. So we want to find new targets and develop antibiotics which target them, right?
Yeah, but the problem, as we'll see here, is that this is the only one that they found that was susceptible to the antibiotic that they screened for was shared by E. coli.
Yes. So that begins the story, right?
I think it does.
So they said one of the reasons they did this because the genome of Wolbachia was determined, right?
That's right.
The genome sequence.
Correct.
Of Wolbachia. So
they probably did it by accident when they ground up these worms and got everything.
So. They said, what genes here could we use as a target?
Yeah.
And they picked this gene called FTSZ.
They picked all the genes that have something to do with cell division.
Yeah. Why would they do that, Dickson?
Well, because they want to stop it from dividing.
Bacterial cytokinesis has emerged as a major target for novel antibacterial drugs.
Right.
What is cytokinesis?
Well, cyto means the cell, and kinesis means movement. So I guess the movement of cells, the division of cells, the way that cells divide.
Cell division.
Cell division.
Many proteins involved in that.
Many.
And one that's been very well studied in E. coli is FTSZ. What are we going to call it? FTZ? FTZ.
Okay. That's right.
FTZ is involved in cell division. It assembles into the contractile Z ring and coordinates more than a dozen other cell division proteins at the mid-cell site of the closing septum. Remember, when two cells divide in E. coli, a septum forms in the middle and eventually pinches off, and boom.
Then you got two.
And FTZ is a major protein in that activity.
Fantastic.
It also happens to be a GTPase. What does that mean to you, Dickson?
It dissolves GTP.
It uses energy, right?
Oh, yeah.
To do something.
Yeah.
Probably its role in this cell division requires energy, right?
Makes GDP out of it, I presume.
So what do you think of using this as a target?
Well, if it's not in the host, but it's in the bacteria, then it's a great idea. But
Wolbachia. But what if it's in E. coli? Can you get something that's specific for Wolbachia and not E. coli?
The hope is that you could, but...
Good answer.
Thank you. There's another way to look at this too, Vince, and that is, where does the filarial parasite live?
Ah, that's a good question. In the lymph nodes.
And?
The lymph ducts.
And?
Nature.
No. What about its offspring?
I don't know. Tell me.
The microfilaria are circulating in the blood. Okay, so they're in the blood.
Microfilaria. Okay.
That's right. So the adults live in the lymph vessels, and the larvae that they give birth to, that the insect now has to pick up to complete the life cycle, lives in the blood. And where does E. coli live, Vince?
Gut.
That's right.
If it's in the blood, you're in trouble.
So maybe we could use an antibiotic that would, under other circumstances, kill E. coli, but it could never get to E. coli because we gave it in a different way. And then it only can get to the filarial parasites.
Yeah, but you'd have to inject it.
I know.
I don't want to do that to you. Wouldn't you like to take it orally?
What if you wait? I'm working on this. No. Where does E. coli live?
In your gut?
Where in your gut?
That I don't know.
How many parts to your gut are there? Let's start with the mouth.
Billions and billions.
Stop. No, come on. Vince is just waxing silly right now because I've been away for a week.
Billions of bacteria.
No, I'm talking about your gut. I'm talking about the parts of your gut.
All right, we start at the mouth.
Mouth, esophagus.
Throat. You pass the throat.
Okay, the pharynx.
Esophagus. Stomach. Which is divided into parts also, but we won't go into that.
That's true.
Duodenum.
Yes. No, then the small intestine.
All right, small intestine.
3 parts of the...
Duodenum.
Yes.
Fundum.
No.
Ileum.
Do we fund them? And how?
Duodenum.
That's the NIH talking.
Now you're being silly.
I try to be silly sometimes. The NIH is Dewey, Fundum, and How. So you've got the junction between the stomach and the small intestine.
The jejunum or the duodenum?
That's the duodenum.
Got it.
Then there's the ileum.
Okay.
And the jejunum.
Okay.
And that's
I'm a virologist, man.
That's okay. That's the majority of the length of your intestinal tract. Then you've got the cecum, which we would call the appendix.
That's where the appendix is.
And it's the junction between the small and large intestine.
Got it.
And now you have the large intestine.
And then you have a rectum.
You're darn right. Rectum darn near killed him. We'll go back to that later. But I want to ask you a question then, Vince. Where does E. coli live in the gut tract?
Large intestine.
Correct. So what if...
On the walls, right?
Well...
It's in the feces, correct?
It's yes and no. It of course, it has attachment mechanisms to stick to cells. But you're right. It's mostly forming the fecal mass. But there are 12 or 1,500 different species that do that in the large intestine only. For the most part, the small intestinal gut tract is sterile. There are some bacteria that live in the stomach. We know that Helicobacter pylori lives there.
Right.
And sometimes it's a natural inhabitant, right? And other times it's a pathogen. So the point is, what if we could...
It's always there.
It's always there, though.
But sometimes it can hurt you.
Correct.
When you get older, you get ulcers and gastric cancer, yeah.
That's right. So what if we could design a drug? Maybe we could design a drug that is absorbed only in the duodenum.
No, it's too hard, Dickson.
No, it's not too hard.
Too hard. You want to put a capsule on it so it only dissolves in the past the stomach, in other words.
No, it's a pH-sensitive absorption mechanism. So if
you're getting complicated.
Come on, life is complicated.
Can we just design something that doesn't hit E. coli?
Not if they share the same gene.
Maybe it's different enough so that you could find a chemical
I don't think so. I don't think this is an essential gene necessary for cell division. You're not going to get around this one, kiddo.
I bet you would. Look, if you're my boss and I'm at a pharmaceutical company and I say, Dickson, I want to do this screen to distinguish E. coli from Wolbachia fitzi, what would you say? No, you can't do it?
Well, the interference part of this antibiotic hits the pocket for the GTP binding.
Well, it depends how conserved it is.
It's very conserved. That's what they said.
How about if I propose to do the structure of both proteins first?
That's a very expensive project.
But you're a big company.
Why don't you just sequence it?
It is. That's, in fact, how they found the Wolbachia.
I know that. But the drug itself... It blocks the pocket. That's how it interferes with the action of this drug, of the GTP.
Well,
ACE.
All right. So you have one way, and I have a different way. But that's the way science goes. We have two different ideas, and mine is going to go forward, not yours.
That's not true at all. Here's the idea that
I'm just kidding. I'm making a joke.
I know. But in the drug companies, that's not even important. The important part is to do it the cheapest you can and make the most money back for it. That's their mantra.
Okay.
All right, so I would say whichever method is cheaper to pursue and results in the same thing, go for it. And then that's up to you to figure that one out.
So this protein was selected because it's been studied extensively
in E. coli.
There are many other proteins involved in cell division, but this one they have the most experience with this. So they found it in Wolbachia.
But they didn't find the others from E. coli in Wolbachia.
They did not.
Isn't that unusual, Vince?
Why don't they pick one of those to inhibit?
So, wait a minute. Maybe there's a reason why they didn't find it.
I can think of a reason.
What is it?
Because they're not there.
No. I know they're not there, but why aren't they there?
Evolutionarily lost?
Perhaps. Because...
It's an endosymbiont.
There you go.
Is E. coli an endosymbiont? No.
It is not.
Yeah. So this guy has gotten some genes from the host.
And it has other genes.
You know, I said something stupid earlier, just 5 minutes ago.
No, you didn't.
I said, they didn't find a lot of genes in Wolbachia that are present in E. coli. And I said, why don't we use those as a target? That was stupid because they're not there. That's the reason.
That's true. But Wolbachia produces substances that the worm needs.
That's correct.
So the worm doesn't have genes for those things.
Yeah, but humans shouldn't either.
That's really interesting. Well, we don't have a gene for vitamin K.
Yep.
Where does that come from?
E. coli.
Thank you.
Or some gut bacterium.
Oh, you're right. I'll accept that.
All right, so we picked this gene because it's there. We know it's homolog in E. coli. All right, so Dickson, you're the head of this program at Dixon Despommier Enterprises, LLC.
DDD.
And we have done this Wolbachia sequence,
Yep.
and you've identified FITC, and we want to use it as a target to inhibit Wolbachia. What's the next step? What would you do?
A drug screen, Vince.
Against what?
Against the pocket.
Where are you going to get this pocket from?
Well, the pocket's there. It's in FITC.
Okay, but where are you going to get the protein FITC?
I'm going to clone it.
Exactly. And then when you got the DNA cloned, what are you going to do with it?
I'm going to express it in E. coli.
That's right. That's what they did. They expressed FITC
Wait a
in E.
minute.
coli.
Why don't we just express the E. coli gene and get it over with? No, you can't do that.
Because
You could do that. You could do that.
I say that they're different enough that the screen would be different, but you don't believe me.
Well, you know what? It's a pretty easy thing to do, so I'd say do
Look,
both of
you'd
those
be
things.
fired if you proposed to screen for anti-Wolbachia drugs using an E. coli enzyme. You'd be fired.
No, they'd never know. I'd never report it.
Well, that's duplicitous, Dickson. You can't do
Duplicitous?
that.
I could
Yes.
do them in two different places?
All right. They expressed
Okay.
protein.
They expressed the Wolbachia FITC
In E.
in
coli.
E. coli and then got a lot of it to
And
work
what
with.
was cool was that it didn't bother E. coli.
Isn't that weird?
Tells me something.
What does it tell you?
It's not interfering with the FITC in E. coli.
Well,
It could, if it were different enough, it could take some of the partners away from the reaction in E. coli, but it's not doing that.
no, but it could have done something else. Maybe there's an upper limit to the amount of FITC that you need to divide, and if you exceed it, it doesn't really matter.
Okay. Then they said, is there an activity to this protein that
They
we can
did.
assay?
They did say that.
And they must have had precedent from E. coli.
That's right.
And that's where they said, this recombinant protein has GTPase activity. So the E. coli protein is a GTPase as well. So is the Wolbachia protein.
So it was folded properly, and the red amino acids were present, and now they have a valid target for drugs.
And that's what you could use for your screen.
Here, here.
But before doing a screen,
Yes.
they did an experiment using something called berberine.
Correct.
Do you know berberine, Dickson?
Well, until I read this paper, I wasn't
You'd never heard of
familiar.
it? It's a plant alkaloid.
So it
What's a
is.
plant alkaloid?
It's a very unusual, complex biochemical produced by a wide variety of plants, as I've found out, just doing some peripheral reading on this, that have antimicrobial activity.
You think they're defensive for the plant?
They just might be, Vince.
So the berberine inhibits the GTPase activity of FITC.
Right.
Why did they do that?
To see if they could then take the next step and use it as an antimicrobial agent against the Wolbachia inside the parasite.
And how would you do that experiment?
Well, I need to get some parasites first.
Right, so if I wanted to get Brugia malayi,
Yes.
like here I am in my office in New York, how would
Yep.
I get some?
I would go online to the NIH grants listing, and I would find out who's working on Brugia malayi. I would then email them and ask them to send me some gerds.
Gerds?
Gerds.
What's a gerd?
Gerds. A gerd is a gerbil-like animal that lives in the desert.
Oh, really? That's what you grow these in?
Yeah.
You can't grow them on a petri dish?
Gerds.
You can't grow Brugia
Gerds.
in a dish? Really? You need an animal?
You need a host.
All right, you're going to get some gerds, and they'll send you some Brugii as well?
Some infected gerds.
No, and then what do you do with the infected gerds?
You dissect out their lymphatics and get out the adults.
Yeah.
And you could also bleed them and then concentrate the microfilaria. Now you have some parts of this to work with. If you had an insect colony of the right vector species, you could actually complete the life cycle in the lab.
But they say here they take adult worms and
They
they
did.
add this berberine to them,
They could do that.
and it inhibits their motility.
Right.
What does that mean? How do you measure motility?
Well, you observe them under a microscope.
Have you done this with worms?
I have.
And you can put a number on this motility inhibition?
It's not easy. But if it stops moving, of course, it's all or nothing.
Could you measure how far they go? It says they show no movement.
Vince, this is a side issue, but let's take it for a moment, okay?
Yeah, we can go side.
I think in TWiP 101, we haven't done a 101 that's been as inclusive as what we're up to now. Parasitic nematodes. Okay, the nematode family is huge. There's an enormous number of organisms that are considered nematodes. Okay, and there are only two kinds, right? And what are those two kinds, Vince?
There are only, repeat the question.
There are hundreds of thousands, probably, of different kinds of nematodes in the world, and there are only 2 kinds of them.
That are parasitic for humans.
And non-parasitic. Those are the 2 kinds of worms.
You're not asking me. You're telling me.
Well, I was trying to drag it out of you. But, okay, so you divide the world up into 2 parts.
Parasitic and non-parasitic.
You're either a parasite or a host.
So C. elegans is a non-parasitic
correct.
nematode.
That's
right?
correct.
Okay.
That is right. So let me tell you what happens then when you watch them move. They will all move the same way. I'm doing this motion with my hand there. It's called serpiginous motion.
He's making his hand is a snake.
That's right. And like a serpent.
And
okay?
Dickson is good at that because he is a snake.
Now that's what happens. Stop it. That's what happens when you take these worms, I should say, out of soil and isolate them and just observe them in just distilled water. They will
undulate.
Undulate. Good word. Undulate. If you take trichinella from the nurse cell that's all coiled up inside and you digest away the tissue and release the parasite,
yes, it
doesn't do this at all. You know what it does?
What?
It coils and it uncoils. It coils and it uncoils. That's all it does.
We talked about that, yes.
Right. But if you remove a certain coating from the outside of that stage, they will assume a serpiginous motion. I don't know if we talked about this part or not, though, because this is the part I wanted to get to. I'm going to imagine now that your table is a shallow tank of water. We have one end over here and one end over here to my left. On my right-hand side is the hottest end, and on my left-hand side is the cooler of the 2 ends. And in the middle is a neutral temperature. Let's say on my right hand it goes up to 38 or 40 degrees centigrade. On my left hand it goes down to 10 degrees centigrade. And in the middle it's like 20 degrees centigrade. Now, with equal distance between 20 and 30 and 20 and 10. Now you take a free-living nematode. Free-living meaning non-parasitic nematode. Okay, any free-living nematode of the hundreds of thousands of species. And you put them in the middle at the 0-point gradient or at room temperature of 20 degrees centigrade. I'm going to ask you to guess at which direction the worm will move. To my left or to my right?
Warm or cold?
Right. Left is cold, right is warm. A free-living nematode put in the middle of the plate.
I would say warm.
Thank you. Now I have a parasite. Let's say I take the Microfilaria of Brugia malayi, and I isolate them from blood, and I get them into a nice little pellet at the bottom of the tube, which I can do easily. And I take a pipette, and I pipette these worms to the center of the plate at 20 degrees centigrade, and here's the cold end, and here's the hot end. Which side do you think they're going to migrate to?
Cold end.
Vince, you got it wrong.
Well, I could tell you were setting it up, but
I was setting it up. You have to reverse everything you just said.
All right, why?
Well, I can tell you why.
They both go to the warm end?
No, reverse. I said reverse.
I said cold.
No, you said hot for the free living.
But you said I was right when I said hot.
I didn't say you were right. I gave you the next choice.
Okay, cold for the free living and hot for the Microfilaria.
Correct.
The Microfilaria want to go to the liver?
Or to blood, or to the host.
But why cold for the free living?
Good question.
Because they don't need to be inside anyone, right?
Bingo.
Yeah.
And this is true for all free-living and all parasitic nematodes. They will migrate. If they're a parasite, they'll migrate to the hot end. If they're a free-living nematode, they'll migrate to the cold end. Now, what's even more remarkable, Vince, is that the parasitic nematodes, if you place this gradient at too high a level, and it exceeds their tolerance, and it's lethal,
yes,
they won't care. They'll migrate right to the end. And
you sure?
And they'll kill themselves. They have no way of preventing themselves from migrating to a certain point and then stopping.
Yeah. So this is a way to get migration of worms, you're saying?
Well, no. Then all I'm saying, I said it was a sidebar. This is a sidebar,
I'm wondering what this has to do with that.
It's a sidebar, but you can observe them moving, okay? So you can test to see how far they can move on both sides of this,
and
right?
And you could put drug on them if you wanted to,
and they didn't do that. All they did was just observe them in a petri dish and then saw whether they moved or not.
Yeah, let's see how they...
They didn't measure the thermal gradient response.
No, they're just measuring motility.
Yeah.
Percent motility.
It's easy to tell. You know,
They also look at making microfilaria.
Yes.
So they put the drug on it stops them from moving, and it also says it inhibits the production of microfilaria. Can you explain that, Dickson?
I can. I'm a little bit concerned about the word production rather than the shedding.
That's probably what they mean, right?
I think they mean shedding.
Yeah.
Because they're already produced inside the worm. Okay, the uterus of the worm is loaded with microfilaria.
Mm-hmm.
And so what they're looking at is the shedding of microfilaria.
Yeah.
And so this drug probably slows down or stops the process by which microfilaria are born. Mm-hmm. That may be an active process on the part of the microfilaria, or it might be a peristalsis on the part of the uterus of the worm to squeeze the parasites out the vulva.
Well, it could be if this thing paralyzes the worm. That's an effect.
It'd
You
be
get rid
good
of
enough.
motility and you get rid of squeezing out.
That's correct.
How do you measure microfilaria production? Can you see them, or do you have to centrifuge them away from
No,
the
they can be
adults?
seen. They can be
see them?
seen. And so in a peripheral blood sample that you get from an infected GERD, you would take a volume of blood in a microcapillary so that you know what the volume was, and you know what the volume of blood is in a GERD. So
Right.
you measure how many microfilaria are present in this microcapillary worth of blood, and you multiply up to see what the production level is.
But these are done outside.
This is done in vitro. This is done
So
in
how
vitro.
do you measure on a plate where you've got these worms sitting? How
Just
do you
count 'em.
measure? You
Just-
look at the
just
microfilaria.
count 'em.
So Dickson, I have in my hand a pen,
Yes.
and I have a line. This is a worm, okay?
It is. It is.
How big would a microfilaria be, like that?
It's micro.
Be a point?
You need a microscope. In fact, if we go to the videotape, as Warner Wolf used
Want
to
to look
say,
at your book?
sure, I have the actual size of a microfilaria sitting right in there for you to see. In fact, I photographed them myself.
Pardon me for that clicking and tapping as I reached.
Right. In the back of the book, there's an atlas.
Shrugged?
Shrugged. Atlas shrugged, that's right.
All right. Which figure is it?
Keep going.
Which way?
I
Hot or
don't know.
cold?
I don't know where you are. Hup, stop.
Wucheria. Wucheria.
That's right.
Wucheria?
Wucheria.
3 tries. All right. Brugia malayi. These are microfilaria.
They are. How big are they?
Oh, which one? These are red blood cells, not or white blood cells.
Those are white blood cells.
This is the microfilaria.
Yeah.
Where is the adult worm?
I don't have a picture of it there.
These are quite large.
This
I
is
would
a
think.
diagnostic atlas. So this is the stage that you'd find in blood. No. Look at the size.
200 microns by 6 microns.
That's how big they are.
So an E Coli is a micron, right?
Yeah.
So these are pretty big. So
They're
the
the
adults
same
must
width
be
as
huge.
they're the width of a red cell.
So
The adults are very large.
big.
The adults you can see with your naked eye.
Okay, so you have to put a scope on to measure this. Is that right?
You do. And the way you prepare this is a special technique named after a gentleman whose last name was Knott.
Knott.
That's correct. So this is the Knott technique.
Is he related to Knott's Berry Farm?
No, Vince.
You know what I'm talking about?
Yes, I know what you're talking about, Vince.
You
But
weren't born yesterday, right?
I was born yesterday, trust me.
So we have these 2 assays, motility and
Shedding.
microfilaria shedding. They're both inhibited by this plant alkaloid.
Yes.
And they say they need 20 micromolar. And to do the same with doxycycline, which is a...
Antimicrobial
That's
agent.
right. They only need 10 micromolar, so it's comparable, right?
Yeah.
If you needed 5 millimolar of berberine, that would be significantly more.
I think
right?
so.
Because a millimolar is 1,000 micromolar.
Indeed it is.
That's right. That is a fact that will never change.
That's right. Thank you, Avogadro.
So they can inhibit the GTPase activity of this berberine. The GTPase is measured in a chemical reaction. They can also inhibit, with the same compound, the worm's motility. Does that prove that inhibition of GTPase causes the phenotype in the worm?
Ooh, good question, Vince. No, I think you'd have to do some other studies.
Yeah. You'd have to do other things.
You'd have to use some gerds.
It's a funny name for an animal.
It is, isn't it?
Gerds.
Well, gerbil isn't exactly on the list of most serious names of mammals I've ever heard either.
And the last experiment, they do your drug screen. They have a library of chemicals.
Right.
And they run it past the assay, which is this protein with GTPase activity. They say, what chemicals in this library let's see how many libraries. A small molecule library. I don't know how big it was. It could be 50,000. It could be 300,000.
It's an amazing world out there,
They do a
Vince.
high throughput screen using the assay GTPase activity of FITSI, and they find some inhibitors.
They did.
And that's the end of the paper.
I know.
What would you have done next with these inhibitors?
Ooh, Vince. Take them to the animal
They only
model.
show that they inhibit GTPase.
I know.
What do you have to do next?
Well, I think I would do toxicity studies next if I was a drug company to see if they're harmful to people.
Wouldn't you first see if they inhibit worm motility and microfilaria shedding?
Well, but of course.
They didn't do it.
Well,
Do you know why?
don't know.
They're not soluble.
Oh,
They're not terribly soluble.
strike three.
So they were unable to reach the necessary concentrations to do worm experiments. That's unfortunate, isn't it?
It is unfortunate.
I'm looking.
These are
here.
heterocyclical compounds that look like dihydroxy chicken wire.
Yes, they are dihydroxy chicken wire. I can show you a picture of them here, Dickson.
Yeah, it's in the article. I've looked at them. My eyes tend to glass over when that occurs. But you know what? Ivermectin is much
Here
more
you go.
complicated than that, and that's soluble.
Yeah, they say the solubilities is poor, precluding measurement of IC50 values.
So is there a way to make them soluble, Vince?
Now you have to modify the chemical structure. You put some more dihydroxys on.
That's right.
Onto
You
the
make
chicken
them
wire.
HCLs or stuff like
By the way,
this.
berberine is a dihydroxy chicken wire too.
Sure is.
Should we tell the audience what dihydroxy chicken wire means?
Sure.
Okay, good. I agree.
Go ahead.
It just refers to the fact that these compounds are made of multiple hexane rings, essentially,
Right.
each of which looks like a segment of a chicken wire.
Correct.
And chicken wire fences, right?
Correct. That's right.
And dihydroxy because it's just
Yeah.
sounds good.
I had the pleasure once of giving a refresher course to pharmacists who had been out of school quite a long time, obviously, and they were frightened that I would just throw up a whole bunch of chemical symbols on the board and start talking gibberish because they wanted to really learn how their anti-cancer drugs worked. And in those days, there weren't that many. And I had a little knowledge of the way DNA replicates, et cetera. So I was going to give them a night course. So I started out with 3 chemical symbols, right, or 3 chemical formula.
Right.
The first one was a capital C-H2O. And I asked people to tell me what that compound was, and they looked at it, and it was a pretty simple compound, C-H2O. And no one could guess, because everybody in the audience was at least 30 years old or older and well out of school. And I said, well, that's the formula for seawater.
Very good, Dickson.
Wait, no one got it. So then I went to the next one.
It's just an impossibility, though.
That's true. Then I went to the next one. Of course it is. Then I went to the next one, which is a bunch of irons.
It's not an impossibility, though. Sorry.
Yeah, it isn't.
CH2 double bond O. What is that?
Ah, that's different.
What is this formula?
Looks like an ethylene.
Well, there's only one carbon.
Methylene.
Methylene. Yeah.
Methylene. I meant methylene. Sorry.
Yeah, you meant.
I meant.
On the test, you get one chance.
No, I crossed it out and went back and went. So the next one was a circle of FEs.
A circle of FEs,
Yes,
of
with-
irons.
with-
4 of them?
With 3 pluses after each iron. All the way around. All connected with lines, of course.
Okay, what's that?
It was a Ferris wheel.
Very good.
That's what the next one was. And you can.
These are probably common chemical
They
things.
probably are common chemicals. And the last
What
one.
is the seawater, right?
That's seawater. Next one is a Ferris wheel, right? And then someone actually laughed at that point, realizing that I was playing jokes on them. And then the final one that I had was a compound that I just kept drawing on the board with benzene rings and N's and C's and all kinds of things. And on one end I put an OH, and on the other end I put an OH. And I said, what's that one? And no one got that right, except that they were in a better mood, realizing that I wasn't going to snow them with a bunch of chemistry. And I said, that one is dihydroxy chicken wire.
That's good.
And that started the course off. And then, of course, then they would listen to what I had to say. So I had to teach them what DNA molecules were and how they replicated, and I had to show the inhibitions. It was fun,
So,
actually.
Dickson, returning to this paper,
Yes.
they identified a bunch of compounds from this library. They had one, what they call a lead compound, and they start to modify it.
Right.
And All they have is the inhibition of GTPase. They said it didn't affect E. coli, and that's probably because it didn't get in.
Right.
It's not soluble enough, so they have to
Right.
work on this getting in, and that's probably why they also don't have the Wolbachia inhibition.
Right.
But do you think this is a promising approach to identifying new antimicrobials against Wolbachia? They have this enzyme which is expressed. They can modify the leads they got by the screen.
Sure it is. But there are other typical proteins that bacteria make that mammals don't make at all.
Many.
That you could aim for as well, like cell wall.
There are tons of proteins.
Right?
Yeah.
Do they have any...
The problem I have with this, Dickson,
please.
all right, they're going to go through a lot of work.
They are.
They're going to get a drug that inhibits.
They will.
They're going to give it to people, and within a year...
Go on. It's be resistant.
Yeah, exactly.
Sure.
So they need more than one.
Although these are slow-replicating organisms.
And they are DNA-based.
And they are DNA-based.
So they make fewer
Vince,
changes.
Wouldn't you rather attack a molecule that wasn't involved in cell division rather than one that was involved in cell division? Wouldn't you rather attack the metabolism of the cell rather than its division cycle?
Well, this is going to stop the cells from dividing. But
I know.
It's not necessarily killing the worms, right?
Not the worms, the bacteria. I'm worried about the bacteria.
The bacterium, it may not kill them. It may stay there.
So there are...
Not that it matters if you kill the worm.
Well, without their bacteria, they die. That's the unusual thing to find out here. Without their bacteria, they die. That's remarkable.
You asked me if would I rather treat a metabolic target rather than a cell division.
Yeah.
I don't have a particular reason to do one or the other. Do you?
Well, maybe the inhibitors would be more soluble because they would resemble substrates.
I think they could make these soluble. A good chemist could add a hydroxyl or a methyl or something.
Well, maybe that's what they're doing now.
Yeah, they are.
I must also add quickly that if I were a drug company and I got it to this stage, I wouldn't publish the rest of it.
I have a friend who used to be in a drug company, and he says if they publish it, they're not doing it anymore. He said this, he would say they're probably not working on it.
No, they stopped
Yeah,
before
they
they
did
got to
stop.
the good part.
They didn't modify it anymore.
Well, they didn't tell you they did, but they might have. Because they're in business to make money, and they don't want to give away their trade secrets. And taking berberine...
It's pretty sad, isn't it?
Well, a little bit.
But we do have a lead structure there, and
this is
anybody else could take it. But, Dickson, in the end, who's going to pay for this?
I'm going to say this is a great neglected disease. Why do you think they call them that?
Nobody pays any attention to them, right?
Correct.
So Gates has to donate money to get this into people eventually.
I would think so.
So these are in New England Biolabs, the Bloomberg School of Public Health, this Worm Institute. Who's paying for this? Let's see who paid for the research.
New England Biolabs could pay for this themselves, I think. Because that's a profit-making.
Tell the audience, if you'd like to know who supported a research, how you do it.
What would you prefer to get funded from? A private organization like New England Biolabs or Merck or Hoffman LaRoche, or from the National Institutes of Health, or from, like the Gates Foundation?
Well, I have news for you here. There's no acknowledgement of support on this paper.
So it's probably New England Biolabs.
Yeah. And these institutes must have some money. Yeah.
Well, this is one way of funneling it off into their own pockets without having to account for it.
Let's do the second paper.
Okay.
This is also in PLOS Pathogens. Not
right.
Also. This is in PLOS Pathogens.
Uh-huh.
Same family of journals, the PLOS, Public Library of Science, open access, but a different journal.
Right.
It's titled, Wolbachia infections are virulent and inhibit the human malaria parasite Plasmodium falciparum in Anopheles gambiae. So they're saying here that Wolbachia has a different effect than Wolbachia in Brugia.
You betcha.
It's harmful, right? So Anopheles gambiae, what's that?
It's a mosquito, Vince.
I know, Dickson, but...
It's an anopheline
what does
mosquito.
it transmit?
By the way, the other mosquito that we talked about in the other paper, which we didn't mention the name of...
I didn't mention it.
No, you didn't. It's Aedes albopictus.
Yeah, but it's not even mentioned in the paper.
It should be. Yeah, it's there someplace.
Because they didn't work on the mosquito here.
Yeah, but that's where the Wolbachia probably came from.
Because here the Wolbachia is in the parasite.
Yeah, but it probably got it from the mosquito.
Maybe.
Because the mosquito that transmits it, the Aedes aegypti, which is the gold standard for dengue fever, transmission, and for albopictus, which is a laboratory-raised vector for work with the complete life cycle of Brugia, they contain a Wolbachia species, which I think is identical to the one inside the nematode.
Dickson, how long ago do you think that, if your hypothesis is correct,
Yes.
how long ago do you think that the Wolbachia went from the mosquito into the Brugia, the filarial nematode?
I think it went in as soon as the mosquito took a blood meal from an animal that had that infection.
Millions and millions of years
Millions.
ago.
And by the way, Anopheline and Culisine mosquitoes, the Aedes is a Culisine mosquito. The Anopheline is an Anopheline mosquito. Those two branches from mosquitoes diverted a long time ago, millions of years ago. So Culisine mosquitoes have Wolbachia. Anopheline mosquitoes do not.
So that happened after the branching.
And if they did, Vince, guess what would have happened to the Anopheline branch?
If they did what?
If they had Wolbachia, according to this paper, they wouldn't be able to transmit malaria.
Yeah.
So maybe the mutant of Aedes, or Anophelines rather, that shed and got rid of their Wolbachia sometime in the distant past became competent vectors for malaria as the result. It's possible to speculate in that direction.
Yeah.
I wonder, when these two branches of the mosquito line were together in the same tree branch, whether Wolbachia was part of their biology. And as they split, this one lost their Wolbachia while this one kept it. And look what the difference is in the parasite transmission cycles now. Totally opposite. Remarkable. Fantastic. Biology is great, isn't it, Vince?
It's wonderful. So is biology. So Anopheline do not have Wolbachia. Aedes and Culisine mosquitoes do.
Correct. And their parasites have it, and these parasites that are transmitted by Anophelines don't.
Now, according to this paper, it is
Yes.
already known
Yes.
that, well, first of all, let me tell you this, and you tell me what this means. It says, in Aedes aegypti, artificial Wolbachia infections suppress diverse pathogens, including viruses, as we discussed a few
For
moments ago.
dengue. Yes, that's right.
Filarial nematodes. Isn't that weird?
That is kind of weird
Now,
because...
is Aedes transmitted?
Oh,
Are they transmitting? Sorry, the filarial nematodes are transmitted by Aedes. Yeah.
by Culisine mosquitoes, and
Culisine,
also
yeah.
by Anopheline, by the way. They're transmitted by Anophelines, but the Anophelines don't pick up the Wolbachia from the parasites. Isn't that interesting?
So why do they say that they suppress filarial nematodes? I thought we just said they're good for the nematodes.
I don't know where that comes from,
Okay.
because I'm under the impression that both Culisine and Anopheline mosquitoes are competent vectors for filarial parasites.
5, 6, and 8. Let's just briefly look at this. Okay, we can't pick it up. And then the other one that's most important for this discussion. It suppresses the avian malaria parasite Plasmodium
Right.
gallinaceum.
Okay.
So Wolbachia suppresses that. Then they say in Anopheles mosquitoes, in which you put somatically Wolbachia, and this is this Wolbachia strain called W. melpop, which we talked about in TWiP. It's been extensively passaged to adapt it to mosquitoes. Many hundreds of passages. This suppresses P. burgii in rodent
P.
models.
burgii.
P. burgii.
Interesting.
So that from that they get the idea that maybe we can use Wolbachia to control these malaria parasites, right?
Yes, but they ran into a little snag, didn't they?
Yes, they did. Now, what is this meant by somatic infections of Anopheline mosquitoes?
Well, as opposed to the germ lines.
What is that? What is it?
Well,
Me,
you've got
Dickson.
2 lines of cells, Vince. You got
Give
one.
me in my hand here. Show me this this somatic cells.
Everything but eggs and sperm.
Okay, thank you. So my hand doesn't have any eggs and sperm.
I hope not.
in it. So this is not germline.
Unless you've been in the kitchen cooking scrambled eggs or
So
something.
if you just put Wolbachia into a mosquito, it doesn't necessarily go into the germline if you just inject it, right?
That's right.
It goes into somatic cells.
And the difficulty with Wolbachia, Vince, is the fact that you can't culture it alone.
That's right. It has to be within a cell, right?
So it's like rickettsia in that sense. You can't culture it outside of its host cell.
So that's the generation of this Mel strain that's been passaged in mosquito cells extensively, and then they injected into mosquitoes, and eventually they got some that would go into the germline. But it was very difficult.
Correct.
But it hasn't been done with anopheline.
No.
And that's a problem, because even if this works well, the Wolbachia works well to control malaria, the obstacle will be, how do you deliver it to the mosquitoes?
That is right.
If it's not in the germline, it's impossible, right?
Exactly.
That's all you're going to say?
No, I think that's the proper way of stating it.
All right.
Except that you can engineer Anopheles mosquitoes to adapt to Wolbachia.
How would you do that?
I would take some somatic cells of Anopheles, grow them in vitro in Schneider's medium that didn't have Wolbachia, and I would take the Wolbachia cells infected from Aedes albopictus,
Right.
that did have it, and I would culture these two cell types together until the Anopheline mosquito cells. I would maybe even try a fusion experiment to see if I can fuse these two separate cell types and get them to make a single cell, like a hybridoma.
It's a tough experiment.
Not so
Don't
tough.
know if it's going to work, though.
Well,
it's iffy.
You'd find out right away, basically, because you have good cell markers for each mosquito species of cells, or you could microinject cytoplasmic extract from Aedes albopictus into the cells of Anopheles gambii, and then just see if they grow up with the endosymbionts in place. And if they did, then I would just inject those cells back into the Anopheles gambii and hope that they locate into the body and begin a line of mosquitoes that have the Wolbachia in place. How did they do that?
Here?
Yeah.
Well, they just injected into the thoracic cavity.
Oh, okay.
So this is a good question. So that's part of the results. They inject mosquitoes with a syringe into the chest cavity, and then they look at the spread of the Wolbachia in the mosquito.
This is in a cell-free extract?
It's in the mosquito.
No.
I'm sorry, what are they putting into? It's just Wolbachia. It's cells.
That they got from an Aedes albopictus?
It's this W. melpop strain.
You can't grow that in vitro. You have to get it from someplace.
Yeah, it's probably from an Aedes strain.
Okay.
Let me look in the methods here, Dickson. It'll just take me a moment.
Good.
Wolbachia culture and mosquito infection. Wolbachia was cultured and extracted from infected Anopheles cells. No, it wasn't Aedes. It was Anopheles cells.
How did they get into the Anopheles cells?
I don't know. You can't just add it, right?
There must be a long line of Anopheline cells that's been grown for years now with this adapted strain of Wolbachia then.
I think that's the case.
That's the deal.
And I think we talked about that on TWiP. Here's the reference. Can Anopheles gambiae be infected with Wolbachia? That's one paper.
Okay.
16, reference
Who
number
wrote that
16, and 36 is the other one. That's Razgon, Wren, and Petridis. And then the other one is survival of Wolbachia Pipientis in cell-free medium.
Right.
So it can survive in medium.
Right. So you can grind up the cells and get the Wolbachia and then try to transfect.
So they take this Wolbachia purified from Anopheline cultures and they inject it into the chest and they watch where it's spreading in the mosquito.
Okay.
It spreads in all cells except the gut and the ovaries.
Right.
So in order to become germline,
it has to get into the ovaries.
And
so
it's
the first thing that happened here is that the insect's immune response went up.
Well, that too. But first they just look at where the Wolbachia goes.
That's right.
And then they look at the immune response. And then, yes, it goes up. They look at markers for the immune system, various proteins. They go up. And then...
Were they looking at heat shock proteins and stuff like this?
Cactus, Caspar, REL2, CEC1, CLIP7A, SNRP6, FBN9, LPR, TEP15, and HSC70B.
Oh.
Okay? Does it make perfect sense?
Yep, absolutely.
These are just protein markers of the immune system, not heat shock proteins.
Okay, that's an innate immune system.
Yes, right. So it goes up initially, and then it goes down.
Right.
So their hypothesis is what, Dickson?
Is that the bacteria, once it established residence in the cells, started to modify the insect's immune response.
To favor their persistence, right?
You betcha.
So you put
Wolbachia into mosquitoes. You have this initial burst of the immune response as the mosquito is trying to get rid of the Wolbachia.
Right, but some of them get engulfed or ingested by phagocytes or by cells that have the capacity for ingesting. And the next thing you know, they don't digest. They include them into their cytoplasm, and the next thing you know, they're dividing.
That's about 10 watt. You know, Dickson, they say, this is funny, however, at 10 post-infection.
Oh.
It's 10 days. They left out days. Can you believe they didn't pick that up?
Eek.
The immune system is suppressed, and they say that correlates with replication of the Wolbachia. So it could be that the Wolbachia is now antagonizing the innate response.
And what proteins do you think the Wolbachia would be giving off or substances which would modify the innate immune response for insects?
It could be
that's a very interesting finding.
any number of things.
I mean, if I was in big pharma, I would look for that molecule rather than worrying about whether it killed.
You'd look for the antagonist?
Yeah.
What would you do with it?
I don't know. I think there's a purpose for this out there. There's a use. You could find a use for this one. You betcha.
Suppression of the immune system to make the mosquitoes die after various infections.
Yes. Because we give them things like BT and things like this to kill them, and they become resistant. Well, maybe giving them this compound would lower the resistance by... Well, that's just the kind of thinking. I don't know if that would work, but...
The next thing they did was to look at P. falciparum in these mosquitoes.
Right.
They say here, after infecting with Wolbachia, P. falciparum Cyst development in the mosquito was reduced. How do you do that experiment? How do you get the malaria into the fly?
That's easy.
How?
Well, you have to have an infected host.
Not fly, mosquito, sorry.
No, I know. It's a fly. It is a fly.
A mosquito's a fly?
That's right. It's a dipteran. It is. It's got two wings. It's a dipteran.
Why do we call them? We don't call them flies.
No, we have another word for
but
them.
It's technically correct.
It is.
Okay.
It is. That's very good,
How do you
Vince.
get malaria into the...
Well, you have to have a host that has the malarial parasite in their red cells. And then you have to allow these mosquitoes to feed on that host.
That's the only way. You can't inject into the mosquito.
You probably could, but
So you
that's
have a
not how
mouse.
it's done.
So P. falciparum will grow in mice?
No.
In what?
It grows in Aeotis monkeys.
So that's how they do this experiment? They have monkeys? Let's see.
You can also have cell cultures of P. falciparum. You can have it, but they're not continuous long-term cultures, at least to my knowledge.
So if you had a culture, how would you get the mosquito to feed on them?
Oh, that would be fairly easy, though. They have good membranes.
They were offered a P-infected blood meal.
Right.
That means an animal, right?
That's what it says.
But it doesn't say what kind of animal. Could be humans, right?
No. Probably not humans.
It doesn't say what animals. They just say a blood meal. Could they give them blood?
Sure.
And the mosquito would drink blood out of a dish?
Not drink. They don't drink it.
Right. I'm anthropomorphizing.
You are. It's an artificial membrane. It's like an elastic membrane of some sort. It's made out of a plastic, as I recall. And they can actually put their proboscis through that, their hypostyle, and suck the blood out from that.
So what is an cyst development, and how do you measure it?
Right. So this takes us back to parasitology 101 again.
It does.
In fact, to our malaria.
Is that bad?
No, it's really good. So when a mosquito takes a blood meal, and the reason why we're calling it a blood meal is because she doesn't just, it's not a he, only the females feed on blood. And they're not using it as food, basically. They're using it to get the protein out from the red cells. The hemoglobin is broken down. The heme part is discarded, and the globin part is kept. And the globin is then digested.
Right.
And the free amino acids go into the hemocoel of the mosquito, which is the open blood space. And the hemocoel, the hemolymph, bathes all of the organs of the mosquito, including the ovaries, and the ovaries selectively take up this abundance of amino acids, and they refashion the amino acids into proteins like yolk proteins, and they make eggs. That's the way the mosquito behaves. So if a red cell that's infected is ingested, the outer membrane of the red cell is digested away, releasing the parasites into the stomach lumen. And there are two kinds of parasites. There's female and male gametocytes. They're called gametocytes, okay, because they're not quite gametes yet. In the blood, they're pre-sex stages. When they're taken up by the mosquito and released into the stomach lumen, they develop into the sexual stages.
Mm-hmm. All right. So if you inhibit oocyst formation,
well, the next thing that happens is that these male and females, one exflagellates, they call it, but they make sperm basically, and each one of these breaks away from the male,
right,
and finds somehow the female and makes a zygote.
Mm-hmm.
Okay, and the zygote is called the ookinete.
Okay.
The ookinete. And the ookinete, if you'll allow my fingers here to represent the columnar cells of the gut tract of the mosquito, the ookinete actually penetrates between the cells, not through them, but between them, into the membrane that surrounds the entire gut tract, and it comes and it rests between the membrane and the cells.
Okay.
That's where the ookinete is. It's a diploid organism.
Right, got it.
But very soon thereafter, it transforms into the oocyst. And within the oocyst, the diploid organism undergoes reduction and division to produce haploid sporozoites. And
So
that's
they're the infectious guys.
that's the infectious stage
So,
for the malaria parasite.
How do you measure oocyst formation?
That's pretty easy. You just dissect out the gut tract of the mosquito and put it under a dissection microscope. And these are quite visible. They're large and they're round. So you can see on the surface of the stomach, you can tell how many there are, and you can count them. In fact, they do this all the time in malarious areas to see what the transmission pressure is for malaria and how many mosquitoes are infected and how heavily they're infected. They can do that just by counting the oocysts in a number of infected mosquitoes.
So if the Wolbachia is inhibiting oocyst, it could be acting anywhere upstream of.
We don't have a
we don't
clue.
know.
I mean,
But
this paper doesn't address that issue,
The
but
point is, you're not going to be making sporozoites.
you're not going to be making
you won't
sporozoites.
the mosquitoes will not transmit the infection. To
That's
the
very
host.
selfish for Wolbachia to do this, right?
Yes. Well,
It's-
this is not normally where you find Wolbachia.
You know what? Because it's a pathogen for the mosquito, too.
Well, that's in fact they found that these mosquitoes with Wolbachia would die if they had a blood meal. So Wolbachia infected mosquitoes were fine unless they had a blood meal, and then they died.
What does that mean?
That I don't know. But they speculate for a page on what that could mean, because they say when you take a blood meal, all kinds of things happen, and that could be intersecting with the Wolbachia, but they don't know. But why is that a problem, Dickson?
Well, Vince, if you wanted to use this as a control measure, it doesn't sound like it's going to work out too well.
Why?
Well, because the females will die and they won't transmit their genes onto the next group.
They won't transmit the Wolbachia, right?
Or the Wolbachia.
Of course, they don't even have it in their germline to begin with. But if you could somehow overcome that, as we discussed, this is a killer. This is a game ender, because the females will die. And they'll
Correct.
never move the Wolbachia. So they have to get around that.
But they only die when they take a blood meal.
Yeah, but don't they always take blood meals?
Well, eventually, yes, but they live for a week, up to 2 weeks before they do that. So this could succeed at some level as a control measure.
Will they mate before they take a blood meal?
They mate before they take a blood meal because that's where they store the sperm. They mate first, almost immediately after hatching, they mate. And then they carry these sperm around with them, and now that changes their behavior, and they start looking for a host to get blood from.
In fact, they do say, Dickson, that so what they've observed is using somatically carried Wolbachia, and they say if stable infections of mosquitoes germline, if they behave in the similar manner with respect to dying when you take a blood meal, this acute mortality phenotype could inhibit the spread of Wolbachia in mosquitoes. They don't say it will. They say it could.
That's right.
So they have to do that experiment, but they don't have it in the germ cells yet. So
No, they don't.
it's very frustrating. They're missing so many things, aren't they?
They are. I'm surprised they haven't tried injecting Wolbachia directly into the ovaries.
Probably have.
Those cells are uniquely different. That is, I'm sorry. Those cells are unique, or those cells are different. We do misuse that word a lot, don't we? So much so that the Wolbachia cannot set up a symbiotic relationship with them. But are they in the germline of the Aedes aegypti?
Yeah,
they
sure.
are. So the yolk material per se is not an inhibitor for their growth.
No.
So there's something unique going on here.
Shall we do a few emails?
I'd love that, Vince.
That was a nice discussion. Did you enjoy it?
Of course I did.
You didn't. Dixon's shaking his head.
Stop.
First one is from Raihan. In TWiP 33, Dr. Despommier said that certain parasites do not need receptors to enter cells, while Dr. Racaniello then said that all viruses require a receptor for entry. I might be wrong, but don't certain paramyxoviruses, like a respiratory syncytial virus, enter the host cell by mere membrane fusion? I hope I'm not missing the point here, but I remember being taught in my undergraduate course that there are a certain class of viruses that don't need a receptor at all.
Vince, take it.
All viruses that infect animals require a receptor. Even the ones that fuse with the plasma membrane, and it's like the paramyxoviruses that you mentioned, they must first bind a receptor, and then they fuse with the plasma membrane. So if your undergraduate course taught you that some viruses don't need receptors, they would be wrong. The only exception is fungal viruses.
Oh.
Certain fungal viruses and some plant viruses that get introduced by insect vectors or mechanical.
Wait a minute. If the virus buds off of a cell and it's now enclosed in the envelope, which is host-derived, has it included a protein into the surface which is specific for the virus, which is the ligand for the receptor?
Yeah, it's a viral glycoproteins get incorporated, yeah.
Okay.
And those are needed to bind the cell receptors, yeah. No, it's absolutely correct that all animal viruses have to bind to a cell receptor.
Like neuromannic
If you'd like
acid
me
or
to
something
write
like
your
this.
professor an email, let me know.
You've written a book, just tell him to go read it.
Steve writes... Steve, this is from Steve.
Okay, Steve.
I first read of the Red Queen's hypothesis in Parasite Rex by Carl Zimmer, which listed Dr. Despommier in the acknowledgments. Did you really?
I did. I had a nice long talk with him.
Wow.
Yeah, sure.
I am just astonished to be in your presence.
Stop it.
Recently, there was a story in the Wall Street Journal, Why Sex to Keep Parasites at Bay, of course, by Matt Ridley. I believe it would be very helpful to hear your thoughts on the Red Queen hypothesis. Thanks. We appreciate all of your great podcasts.
Good. Well, the Red Queen to me says, the faster I go, the more behind I get. The Red Queen is running in place. So it's a paradigm, it's a metaphor for evolutionary engagement between a host and their parasites.
You could say parasite, yeah.
Yeah, they're host and parasites. So but the arms race is a better comparison here than a Red Queen, I think, because sometimes one side gains an advantage and then the other side adapts by being selected for resistant forms. Then the parasite changes again by genetic mutation and random selection that is now able to infect this changed host. And that's how species are created, and that's how new species of parasites are created too. It happens this way all the time. So it's not running in place, basically. It's not running in place at all. It's an advancement. It's a genetic advancement of both the parasite and the host over time.
Now, the parasite sex thing, the idea is
that you get a faster rate of recombination between the same species if you had sex than if you didn't.
And it helps you be resistant to parasites.
Apparently, because it's got a genetic component which says that the more mutations you have, the better the chances are that one of them will be against this parasite. Now, it won't be able to affect you. But I like saying that. But viruses can take cassettes of genes from hosts and give them to other hosts, like from a resistant host to a susceptible host, or from a susceptible host to a resistant host. It can mix and match. It's like, you go down to Midtown and you see these guys with big overcoats, and they throw open half of their overcoat, and inside they've got a bunch of Rolex watches, and they want you to take your pick. And they say, go ahead, have one. And, you can take your pick of, Movado, and they're all made in Nigeria, by the way, and they're all $10. That's strange that works out like that, isn't it? The point is that the viruses can shuttle genes laterally rather than vertically.
So
I think the Red Queen hypothesis here has to be modified.
The basis for this sex and parasitism idea is that some experiments have been done where asexual organisms are easily wiped out by parasites. But if you have sexual organisms, they can be resistant.
That's in a laboratory setting.
Well, this has actually been done in the field. In Matt Ridley's books, he talks about, in The Red Queen, he talks about field experiments that have been done to address this. But if you consider a organism that just undergoes binary fission, which would be more susceptible to parasites because it can't get fresh genetic material, can't they be infected with viruses that would bring in new genes?
Of course they can. I mean, they all have bacteriophages that attack them, don't they? You're talking about binary fission, like bacteria that divide by...
Bacteria, yeasts, any
Yeast are
kind
sexual.
of...
Yeast have sexual stages.
But they also have binary stages, right?
They do. Or you think about protozoans.
Anything that has a binary stage, you could have material being introduced by viruses or plasmids. Naked DNA can get into cells, right? So I'm not sure that without sex you have a complete susceptibility to parasites, because as you said, you have the possibility of introducing genetic material
in another way.
And the experiments that were done in that book. Have you read The Red Queen?
Sorry that
I will bring it in and show you the experiment
in the field
that he's talking about. Yeah.
What organism was it?
I can't remember. It was some kind of protozoan or
I see.
One of these rotifarian-like things.
That's not a protozoan.
Yeah, I know. I said. Look at the word. I don't remember. I'm just talking randomly now.
Got it. I
will bring it in, show it to you, and we can get back to this next time. But I don't think it explains everything. Sex.
No.
Scott writes, Vincent and Dickson, I listened with great interest this afternoon to the TWiP story on malaria vaccine via Science 360. You know, that's the NSF
I've listened to that
radio.
Yes, I have.
I am not an academic, but a true science junkie. My keen interest is due to the fact that I lived in Nigeria for some time and suffered from P. falciparum infection 7 times before I was finally able to build enough immunity to not suffer mightily.
7 times? Wow. So it was a Peace Corps worker, maybe.
No cerebral, thank goodness. But it left me with a passion for watching developments in the study of the parasite and the science of malaria control.
You bet.
And listening to the part on the TWiP podcast about the locus of injection sites being critical to efficacy for this possible vaccine candidate, a thought occurred to me. Since mosquitoes are so adept at finding blood vessels from which to suck their blood meal, why not create a genetically modified mosquito that would produce the candidate vaccine protein in its saliva, so that when it inserts its proboscis into the blood capillary, the host gets a tiny injection of the protein at the same time. While I do expect that not a single mosquito bite would produce immunity, of course, the many thousands that one receives over time might do the job and would maintain it over time as booster shots would be received daily. Since this vaccine seems to be efficacious against all the strains it has been tried against, it would suggest that the parasite's docking protein is highly conserved in evolution, making the evolution of resistance much less likely. If the same genetic sequence were also used to invoke immunity to Plasmodium by the mosquito itself, it would give the mosquitoes an evolutionary advantage in the environment, in that there is also a cost to the mosquito as well as in carrying the parasite. This would ensure that the genetic modification would become dominant in the mosquito population over time. It would also provide a double-pronged attack on the parasite, both at the mosquito and human blood stages of the parasite's life cycle would be suppressed. While I recognize that there are surely some formidable barriers in the way of creating such a system and deploying it in the field, my thought is that it might have enough promise to be worth looking into. Warmest regards from so far malaria-free Costa Rica.
Nice.
So let me just say one thing.
That's a great letter, by the way.
You can't release a vaccine.
No.
You have to have a controlled vaccine.
Correct.
And I don't know how you would make sure this mosquito would drive through the population unless you put Wolbachia in it.
Or a meiotic drive gene.
Yes, so some kind of driver.
Yeah, that's
but
right.
it's Aedes.
Yeah. No.
Anopheles.
Well, you could put it in Aedes then, which never had the opportunity of transmitting malaria.
Yeah, they would still bite you. Right?
They would.
But you have to make sure that the antigen doesn't have a negative effect on the driver.
Another thing is that I agree with the analysis that a single mosquito bite will not result in immunity. But what about too many? You can imagine where this thing might actually, you've had a meiotic driver and you replaced all the other mosquitoes with this mosquito and you got bitten thousands of times a day, which then you might have, that's right, you might have a tolerance effect built up. It's a clever idea. I mean, I think that people who manipulate mosquitoes for these purposes have thought about all of this. Tony James is one of the people I'm familiar with, and Lou Miller and Bob Quads, and Rubiero, Dr. Rubiero down at NIH. There are lots of people out there that are looking at this as possible ways of solving very difficult parasitic disease problems. But there is an ethical issue at stake here, and that is that you're releasing a vector which has competence in other areas, like all the viral infections, for instance, which you could easily do. Well, okay, so...
But that doesn't help people who are going to go from another country without malaria to a malaria country.
No, that's right. So
you have to give them a vaccine.
That's right. That's exactly right. So there's another way to think about this. You know, these pressure injectors, all right? Not the ones that stick right through your skin, but the ones that push it through by force.
Air.
That's a possible delivery system that you could think
Which
about.
more approximates the mosquito bite.
Exactly.
I see.
Exactly.
The next one is from Trudy.
Because actually, we got rid of smallpox by injecting it.
Scarification.
Yeah, we did.
Scratch the surface.
No one had an objection to that one because smallpox is such a horrible disease that this was a risk-benefit worth taking.
Okay. Trudy is next.
Go, Trudy.
Listening to you in podcast number 6 on tapeworms.
Oh, dear.
You are making me laugh. I live in Florida. Not everyone is retired in Florida. Not even people who live in South Florida are all retired. For your information, Whole Foods does list the country of origin of the animal proteins sold in their stores.
Good.
Well, with my background in public health epidemiology and communicable disease clinic, STD clinician, TB outreach, I've never seen a tapeworm in a specimen since moving to Florida. I had seen one in the teaching hospital I worked at in Grand Rapids, Michigan, in a specimen from an immigrant. Now, I said not everyone in Florida is retired, and they are not, but I am retired early.
Right.
Cattle ranches are not just in the panhandle either. Many of these ranches are all over the state. Florida is a big state. We have a lot of open land used for all types of agricultural purposes. While there are beef cattle grown in Florida, many more farms, ranches are devoted to dairy production.
Interesting.
I'm a nurse, an artist, and a Florida master naturalist. Science, microbiology, and infectious diseases continue to be of tremendous interest to me.
Oh, that's great.
We read the rest of this letter on TWiP because it had to do with viruses.
Got it.
By the way.
Oh, nice. Trudy gets a double hit.
Yeah. And the last one is from Charlotte, who writes, in TWiP 33, you asked which protozoa should be discussed in future episodes.
Uh-oh, here we go.
If you discuss Acanthamoeba as a Trojan horse, you can correct errors made on 2 episodes of TWiM regarding Legionella as endosymbionts of Acanthamoeba.
Ooh. TWiM. I'm not a member of TWiM.
Yeah, you like that we made errors, don't you, Dickson?
Thank God somebody else admits to being human.
I think listeners would find comparisons of Acanthamoeba, Naegleria, and Hartmanella in terms of pathogenesis and as bacterial reservoirs interesting.
Yeah, we could do that.
I think we are planning to do this.
We are.
Maybe with Knersh.
That would be good.
Thank you for the podcasts, without which I would never sleep. Charlotte. P.S. The Trojan horse term was coined by Barker and Brown, but everybody uses it now. Do you know this?
I don't.
Do you know what a Trojan horse is?
But of course.
That'll do it for TWiP number
31.
54. You're way
34.
behind.
Sorry, 34.
And you can find TWiP at iTunes, at the Zune Marketplace, or at microbeworld.org/TWiP. We also have an app you can use to stream the episodes to your iPhone or Android device. Go to microbeworld.org/app to learn about that. Also, if you are new to TWiP, please go over to iTunes and leave a comment, because that helps us. There's one reason we want you to do that, and that is so we can get more listeners. Because if you leave a comment, the podcast stays on the front page of the medicine section. More people will see us. We get more listeners, and we spread the word about parasitism, which is what our goal is here. And if you do have questions and comments, we do like to get them, don't we, Dickson?
We love them.
Send them to TWiP at twiv.tv. Dickson Despommier can be found when he's not in the office here
Or in Andros.
Or in Andros at verticalfarm.com, trichinella.org, and medicalecology.com. As always, Dickson, it's been a pleasure.
Vincent, it is. I share that feeling entirely.
Thank you so much. And I, of course, am Vincent Racaniello, and you can find me at virology.ws. I am never an Andros.
Not yet, but I'm working on it.
You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.